Hello everyone. Today I'll be presenting our Intracellular Spatial Transcriptomic Analysis Toolkit. Our toolkit utilizes single molecule resolution technology datasets such as Murfish, Seqfish, and Xenium. And we ask what can such data reveal about subcellular organization and interaction of molecules. In this quest, we define a new statistical phenomena called as decolocalization, whose aim is to find gene pairs whose transcripts tend to be within distance d of each other much more than expected. Our toolkit consists of two primary statistical tests. One is proximal pair test that aims to detect if a gene pair is proximal pair in one cell. And then we have Poisson binomial test that aggregates information from proximal pair test across all cells to detect if a gene pair is co-localized across many cells. For example, our toolkit detected PRK, DC, and FASN to be decolocalized, and you can see that they are proximal pair in many cells. Now we'll give a brief overview of PP test. As we discussed, the goal of PP test is to detect if a gene 1 shown in red color here and gene 2 shown in blue color here, if they are if they are a proximal pair in this cell. So you can see there are 3 transcripts of gene 1 and 2 transcripts of gene 2. So there are 6 possible transcript pairs that may be near within distance t. However, we see only 2 of them. So we ask if 2 out of 6 is significant. So we have a binomial test. Once we have this, we aggregate these results from PP test across all cells. For example, a gene pair I, J is proximal pair in this cell. However, it's not proximal pair in this green cell, and so on. So finally, we count the number of cells in which a gene pair is proximal pair, and you want to ask if this is significant. So we carefully design null model that is not biased towards high gene expression or big cell size. So in a nutshell, I'll describe in different instant functionalities. First, we have different data sets, and you represent these data sets in a data frame format, and you utilize PP test that outputs p-value for each gene pair across all cells. Then we run CPP test to find global map of decolocalization. Next, instant also has differential colocalization that can detect cell type or region or phenotype specific pairs. Instant can also detect spatially modulated colocalization irrespective of spatial annotation. And finally, we have gene module discovery. We ran instant on 5 datasets. Our first dataset was U2S dataset where we detected 304 decolocalized gene pairs at false positive rate less than 1%. And we also showed that instant can detect very reproducible results across replicates and sources. We also showed that instant gene pairs are enriched with geo terms, specifically with molecular function, where we saw that all the instant detected pairs that overlapped with molecular function were annotated with RNA binding. Finally, we showed that instant detected pairs at small distance, such as 300 nanometer, overlap significantly with RNA-RNA interacting pairs, showcasing that RNA-RNA interaction may be one of the plausible cause behind decolocalization. Now we'll show results on differential colocalization. We saw that we can detect cell type specific colocalization across all cell types in our Murfish mouse brain dataset, and UMAP plot of one of the gene pairs specific to inhibitory cell type is shown here. Each orange color here denotes inhibitory cells, while black cross denotes cells in which this gene pair is proximal pair. Next, we do spatial region specific analysis. This is mouse brain, and if you zoom into it, it has 3 regions: CA1, CA3, and DC. And we saw here we show one of the gene pair P-val-B GAD1 that is enriched specifically in CA3 region and whose colocalization in one of the cell is shown here. Next, we have spatial modulation, which can detect gene pair that varies, whose co-localization varies at the tissue level, shown in blue color here. This gene pair is SCK1TTYH2, whose UMAP plot is shown here. Instant can also detect modules. For example, here Instant detected 2 modules that co-localizes with itself. Geo-enrichment of module 1 showed various terms, specifically RNA binding and other binding events related to cytoskeleton. In conclusion, Instant consists of a set of a robust statistical tests to detect decolocalization. You can use differential colocalization module to detect cell type, region, or phenotype specific patterns. In future, Instant may be used to represent a cell as a graph where node represent genes and edges represent proximal pairs. The code is available here. You can check out, and this work is already available on the preprint. Thank you.